Hello everyone, welcome to our YouTube channel. This is the Cell and Molecular Biology lecture series where we embark on an extraordinary journey into the intricate world of cell and molecular biology. In this particular lecture, we will talk about one of the most important issues and topics in the cell and molecular biology, and it is also one of the most pressing frontiers of our scientific exploration, the molecular basis of cancer metastasis. Join us as we unravel the secrets held within cells, exploring the complex mechanisms underlying the spread of cancer throughout the body. Through the in-depth analysis and cutting-edge research, we aim to shed light on this critical aspect of cell biology, offering insights that pave way for new advancement in our fight against cancer. Get ready to delve into the fascinating world of molecular biology and the profound implications it holds for understanding. First is the basic properties of a cancer cell. Well, cancer cells are coming from somatic cell mutation during the lifetime of the affected individual. These are also gene expression in cancer cells which is being altered or being changed. Cancer cells proliferate in an uncontrolled manner, forming malignant tumors. These malignant tumors tend to metastasize, which means establishing more or establishing secondary tumors. There are a lot of massive research undertakings with this, and to improve prognosis, we need earlier diagnosis, treatment of our top cancers all over the globe. We have the breast, prostate, and colon cancer. There are also therapies that lack the specificity to kill cancer cells only, so it also affects your normal cells. Researchers have developed more effective targeted therapies. So you will see here the graph will show you the new cancer cases and the death for breast, lung, prostate, colon, melanoma, lymphoma, kidney, uterine, leukemia, pancreas, oral cavity, and brain cancer. With growth capacity is very similar between cancer cells and normal cells. Cancer cell growth is uninhibited by first is the contact with other cells and all growth factor signals. These cancer cells do not respond to growth factor signals, so they can't stop themselves from growing. Growth potential difference is often attributed to the presence of telomerases. These are enzymes in the cancer cells, and absence of telomerase in normal cells is thought to be a major defense against tumor growth. And also, important reason why we age, why humans age, animals age, because of the declining length of their telomeres, which is being held by your telomerases. So you will see here cell numbers and time in culture days. You have normal cells in negative serum growth factors with positive serum growth factors and negative serum growth factors versus your normal cells with positive serum growth factors. Mutagenic or carcinogenic agents are agents that promote cancer. We have carcinogenic chemicals, ionizing radiation, even viruses. DNA and RNA tumor viruses carry genes whose products interfere with cell growth regulation. Most of the cancer causes and its etiology is unknown. With diet can influence the risk of developing cancer. So this is your the most common cancers in the following population: Japanese. We have first generation migrants, second generation migrants, and Caucasian Hawaiians. So for stomach, more common in male, breast is more common in female, and colon common in male. So these are varying incidence per 100,000. So you will see that stomach Japanese males are more susceptible to the stomach cancer, whereas Caucasian Hawaiians is for breast cancer. Remember the BRCA, BRCA1 and BRCA2. And for the colon males, most of the first, second, and Caucasian Hawaiians have more inclined to get a colon cancer than the Japanese people. Cancer is a genetic disorder. Tumorigenesis occurs by a cumulative progression of genetic alterations. Cells are becoming less responsive to growth regulation and they are becoming more invasive. Genes altered regulate what? Cell cycle, cell adhesion, and DNA repair. Cells responsible for this are the stem cells and your progenitor cells. So this is the oncogenic event A, which affects your tissue or stem cell, which will lead to a tumor subtype X. Again, another oncogenic event A with the pluripotent progenitor cell. It could lead to a tumor subtype Y with your pluripotent progenitor cell will go to the committed progenitor cells, and if another oncogenic event A, it will lead to a tumor subtype Z. Natural selection produces most viable tumor cells, those that does have telomerase expressing cells. Initially, they will be called precancerous cells. So they have loss of certain growth controls. As I said earlier, they have lacked the ability to invade normal tissues or to metastasize to distant sites. With cervical cancer, it progresses over more than 10 years, and they are characterized by cells that appear increasingly abnormal. So you'll see this photo or figures on your right with the cervical cancer. We have your tumor suppressor genes and oncogenes. They're referred to as our brakes and accelerators. With tumor suppressor genes, they encode proteins that restrain or stop the cell growth. Both cell gene copies must be altered. With oncogenes, these encode proteins that promote loss of growth control. They are first discovered in the genomes of tumor viruses. Once they are altered cell genes, we call them proto-oncogenes that act dominantly. So this is your chromosomes, diploid copies, and then there's normal cell growth in blue. One is mutated tumor suppressor gene. As we go further with cell division, two of these cells have red copies. One is a homozygous for the copies of the mutated tumor suppressor gene, which is present in both homologs. And once it's being growth in a dish, it has exhibiting loss of growth control. So how do we convert the proto-oncogene? There's a gene mutation to alter the normal gene product function. So when there's a gene duplication with product overexpression and also chromosome rearrangement with changing gene proximities that alters the product expression level or the function. So when encoding protein with altered structure or function, these have undergone this mutation or deletion. Others are they underwent gene duplication. With this C is they undergone a DNA regulatory sequence that are translocated from distant site that alters the expression of your downstream genes. So they have encoded a synthesis of these encoded proteins. Once they are becoming altered, we can stop them from producing a lot of proteins. Okay, these proteins are mutated and they have been altered. Most of these proteins encoded by the tumor suppressor genes act as negative regulators of cell proliferation. Products of tumor suppressor genes also help us to maintain genetic stability, which may be a primary reason that tumors contains such an aberrant karyotype. So these are the genes with their primary tumors. Like for APC, it's colorectal. Their function is to bind the B-catenin, acting as a transcription factor. And the inherited syndrome is the familial adenomatous polyposis. So for BRCA1, the breast cancer, with the proposed function is DNA repair, and the inherited syndrome is familial breast cancer. So there's a lot of diseases here in inherited syndromes, which mainly associated with the RB gene or the retinoblastoma gene. The retinoblastoma is inherited in certain families and it occurs sporadically in the population at large. Genomes have a deletion in one copy of the RB gene inherited. Development of the RB requires both copies of the RB to be altered or eliminated. Reintroduction of the wild-type RB gene allows a suppression of a cell's cancerous phenotype. So in letter A, you will see here retinal cell with RB genes. Both of them should be affected. So on the third cell division, you will see that the spontaneous mutation in the second copy of the RB gene will lead to the loss of growth control. When in the second figure will show you here that if one is mutated RB gene inherited from the parent, as it duplicates, it undergoes cell division. One is having normal cell growth, and once again it's homozygous. Okay, the mutation in the second copy of the RB gene will lead to loss of growth control. The protein encoded by the RB gene, or we call it the PRB, regulates the G1 or the GAP1 to S transition. So if you have questions about this particular topic and portion of the discussion, you may watch the cell division, cellular reproduction video as we guide you to the process of cell cycle. Transcription factors of the E to F family are being targeted by the PRB. The rest of the cell cycle in the first gap required for normal cell differentiation, and it's being directed by your PRB. Animals with one mutated copy of the RB gene have an elevated risk of developing cancer. So once you are heterozygous for the RB gene and its mutated copy, then you got a high chances of getting into developing cancer. So because the S phase is the synthesis phase, it must be highly regulated by the CDK complexes, okay, the cyclin-dependent kinases, and these transcription factors are being targeted by the protein RB gene. The TP53 is the most commonly mutated gene in human cancers. This is recognized as the tumor suppressor gene that, when is absent, causes inherited disorder called the Li-Fraumeni syndrome, and this disease has a high cancer incidence. Tumors composed of cells with TP53 mutations have poorer prognosis than the wild-type TP53 gene. Loss of TP53 function critical in progression towards malignant cancer. So these are the percentage of mutations of TP53 gene. As you will be showing here, the pancreatic cancer is on our top list, ovarian cancer, the next colon, lung, liver, stomach, kidney, prostate, breast, and lastly, your skin cancer, we call it the melanoma. So the P53 protein suppresses the formation of tumors, and it also maintains genetic stability. So proper functioning of the P53 protein is very sensitive to even slight changes in the amino acid sequence. After the DNA damage, P53 alters the expression of genes involved in cell cycle regulation, even apoptosis or in senescence. The 6 most common mutations in human cancers all map in the protein region that interacts with your DNA. So your DNA here is the double helix structure, and the protein here that has sequences or regions like R175, G245, and etc. is the one interacting with the DNA. And once it's mutated, cancer is most likely to happen. P53 protein acts as a transcription factor, activating expression of a gene that stops the G1 to S transition. And when a cell sustains DNA damage, the concentration of P53 rises so the damage can be repaired before initiating DNA replication. So in general, your P53 protein triggers apoptosis or cell death in cells whose DNA is damaged beyond repair, like your radiation, your ionizing radiation for particular, and all other carcinogenic agents, whether physical or chemical agents. So this is the model for the P53 function. You'll show here the normal cell division is P53 independent in your left, but P53 is required upon DNA damage for repair in the middle, Mutations can accrue upon DNA damage in the absence of p53, which is shown in your right. Senescent cells can remain alive and metabolically active but are permanently arrested in a non-dividing state. One pathway leading to senescence involves expression of tumor suppressor gene INC4A, encoding two separate tumor suppressor proteins. We have P16, which is an inhibitor of cyclin-dependent kinases, and the ARF, which stabilizes the p53 by inhibiting MDM2. The precise role of p53 in directing cells towards the senescent state remains unclear, but inactivation of TP53 gene within your old cells can cause the cells to resume their progress towards full malignancy. Now we move forward with the oncogenes. They are derived from proto-oncogenes. They are first identified as a part of the genomes of RNA tumor viruses, but more have been identified in the laboratory animal models or human tumor samples. The oncogene mutated are most frequently in human tumors is the RAS or the RAS. RAS mutants encode a protein whose GTPase activity cannot be stimulated, which leaves the molecule in an active GTP-bound form, sending continuous proliferation signals. We have here the simian sarcoma virus, which contains the oncogene or the cis derived from the gene encoding growth factor PDGF. The oncogene ERB-B directs the formation of altered EGF receptor. Then it stimulates independently of the presence of the growth factor. Some malignant cells contain excess surface receptors, which increases sensitivity to low concentrations of growth factors. Oncogenes that encode cytoplasmic protein kinases, RAF, a serine threonine protein kinase in the MAP kinase cascade that can be converted into an oncogene, so activity is always on. The oncogene product SRC is a protein tyrosine kinase which phosphorylates proteins involved in signal transduction, control of your cytoskeleton, and even cell adhesion. With oncogenes that encode transcription factors, a number of oncogenes proteins that may act as transcription factors. MYC protein stimulates the cell to re-enter cell cycle from the gap 0 stage. Overexpression of MYC may cause the cells to proliferate uncontrollably. MYC regulates the expression of your telomerases. Oncogenes that encode proteins that affect the epigenetic state of chromatin. Several oncogenes encode proteins that affect DNA methylation or some histone modifications. These critical modification sites are promoters and core histone tails. Oncogenes that encode metabolic enzymes. Tumor cells are more reliant on glycolysis compared to normal cells. So sugar is one of the most important diet for our cancer cells. TCA mutations promote abnormal metabolites affecting histone demethylation, DNA methylation, and resulting in aberrant gene expression. Oncogenes that encode proteins that affect apoptosis. Alterations decreasing your cell's ability to self-destruct, increasing tumorigenesis risk. Overexpression of PCL2 suppresses apoptosis, allowing abnormal cells to proliferate into tumors. Tumor suppressor gene and proto-oncogene products regulate pathways, including apoptosis or the sensitivity to apoptosis after an insult, proliferation and the response to external stimuli, immortalization through the regulation of telomerases, and senescence where cells are metabolically active but are non-dividing. So you'll see here growth factors, for example, receptors for growth factors like the EGFR or insulin receptors. We have your different proteins here, P10, P13K, PKB, RAS, NF1, MYC, MDM2, the RAF, okay, and their effects like immortalization, senescence, proliferation, and apoptosis. The mutator phenotype, which mutant genes involved in DNA repair. Mismatch repair defects predispose cells to abnormally high mutation rates, which increases the risk of malignancy. A deficiency in the nucleotide excision repair or the NER system may be involved in a hereditary syndrome, xeroderma pigmentosa. A deficiency in an NER is implicated in hereditary colon cancer. Tumor cells of this form of cancer often have variations in the length of microsatellite sequences due to errors during replication. We have your microRNAs as a new player in the genetics of cancer. Two miRNAs inhibit expression of mRNA that encodes anti-apoptotic protein BCL2. So it stops the expression of anti-apoptotic proteins. Some miRNAs act more like oncogenes than tumor suppressors. One cluster of your miRNA genes is overexpressed during the formation of some lymphomas. It is being confirmed through mouse studies to act as oncogenes. Some of your mRNAs are implicated also in tumor metastasis. With the cancer genome, hundreds of different genes have been identified as cancer genes. They are thought to have some causal role in the development of at least one type of malignancy. The results of studies of cancer genomes suggest that the same tumors from different individuals express divergent combinations of genes. Number of genes are mutated or few in large proportion of tumors, yet each type of cancer has its own characteristic complement of frequently mutated genes. Colon cancers have a high mutation rate in a small number of genes. Progression of large number of colon cancers occurs from a series of mutations that lead from a benign state to malignancy. About 70% of colon cancers exhibit chromosome instability and aneuploidy. So for a normal epithelium, we have a PC mutation. It leads to hyperproliferative epithelium. It will lead to formation of small adenoma. With KRAS mutation, it will lead to a large adenoma. With the loss of 18q, SMAD4, CDC4, and TP53 mutation, thus leading to colon cancer. The cancer genome, with its mutational landscape studies, we have studied 3,000 tumors, 12 major cancer types, 127 gene mutations, and it's also impacting a core set of 20 cellular pathways or processes. A large number of cancer genes are components of a small number of pathways. Cancer can be thought of as a disease of aberrant cellular pathways. So it's showing here different genes and types of aberration with like transcription factor regulator, histone modifier, genome integrity, RDK signaling, cell cycle, MAPK signaling, PK31K signaling, TGF beta signaling, WNT to beta catenin signaling, histone proteolysis, splicing, HPPO signaling, DNA methylation metabolism, NFE2L, protein phosphatase, ribosome, TOR signaling, and others. With DNA microarrays or using DNA chips, they are used to diagnose cancer. DNA is corresponding to thousands of different genes spotted on a glass plate so that each spot corresponds to a different gene. Fluorescent complementary DNA is also prepared from cancer cells, allowing to hybridize with the spots. Spots corresponding to actively transcribed genes fluoresce. So these are indicated with the colors shown here, like low, dark blue, indigo, and lighter colors for normalized expression, and for the red ones are high mutation rates for different types of genes encoded by your DNA chips. With engineering linkage, we have therapeutic radiation. 60% of cancers are treated with radiation, often in conjunction with drugs, surgery, and immunotherapies. We have what we call as your external beam therapy. X-rays focus on an internal tumor. This is a commoner approach. Rays may converge from multiple sources to protect normal tissues. Internal therapy places an isotope near the tumor's DNA strand. Strand breaks and apoptosis will kill your tumor cells. Conventional cancer therapies may be replaced by targeted therapies based on the molecular basis of malignancy with normal cells undamaged. Therapy targets protein whose inactivation leaves the cancer cells unable to grow or survive, targeted to the cancer cells of a particular patient based on their unique pattern of somatic mutations. There are a lot of new strategies, including antibodies against tumor cells, inhibition of cancer-promoting proteins, and preventing the growth of blood vessels that nourish your tumor. We have your passive immunotherapy that treats cancer with antibodies, AB, often monoclonal, or MAB in origin. Herceptin is humanized monoclonal antibody blocking growth factors that stimulate proliferation of breast cancer cells, or the anti-HER2. Rituxan is an antibody that binds to cell surface protein CD20 of non-Hodgkin's lymphoma. There are other targeted receptors like your EGF, VEGF, CD52, CD19, and CD3. With immunotherapy, you may watch the lecture video for the immune response. The antibodies are the front line of your immunological theory. With immunotherapy, there are active immunotherapy that boost patients' immune defenses. There are a lot of strategies, including targeted immune cell dampeners, your PD-1, PD-L1, removing the patient's immune cells, reintroducing them after stimulation or the prevenge. Concerns remain regarding expenses, toxicity and side effects, and even long-term effectiveness. These are the inhibiting drugs that of your cancer-promoting proteins. Your Gleevec, Tasigna, Spiracel, Iressa, Tarceva, Zactima, Sutent, Votrient, Tycurv, Nexavar, Zilbaraf, GDC 0973, Velcade, and Carfilzomib. Inhibiting the activity of your cancer-promoting proteins. Studies with protein-inhibiting drugs have shown moderate success in most cases. Genetic complexity of tumors and resistance development are rare factors. Best results for chronic myelogenous leukemia CML with Gleevec. Metastatic melanomas harboring a mutant BRAF or the BRAF V600E have an altered active site inhibited by your drug Zelbocaf. With the concept of cancer stem cells, new research suggests that cancers possess stem cells with enhanced growth potentials. Unlike chemotherapies that target these cells, treatments will need to be long-term like Gleevec. Cancer stem cell targeting drugs like Taraxtuma can bind Notch family of proteins, prompts cancer cell differentiation, and it also increases effectiveness of other chemotherapeutics. Lastly, we inhibit the formation of new blood vessels or angiogenesis. This is the formation of new blood vessels. As a tumor grows, it needs more blood supply. Compounds that inhibit angiogenesis are promising treatments for tumors. With interruption of your vascular endothelial growth factor and vascular endothelial growth factor receptors interaction, we use Avastin, which is a monoclonal antibody. An angiogenesis inhibitor denies the tumor access to nutrients and oxygen needed to grow. So this is the cancer metastasis. Thank you so much for watching, and I hope I see you again for our next lecture YouTube